Hello, welcome. Thank you for coming to our second to the last colloquium talk. Don't forget that next week we have one because of the first one from January, the ice storm apocalypse, whatever. That's next Monday, so don't forget about that. But today we have Dr. Kaitlin Casaletto with us today. Very excited to hear about her work. She's a clinical psychologist, neuropsychologist. She did her training in the joint UCSD SDSU doctoral program in California, followed by a clinical fellowship in neuropsychology at UCSF Memory and Aging Center, now the Fine Memory and Aging Center. Congrats! Recently endowed that center in the Department of Neurology. She's now currently an associate professor of neurology at the UCSF School of Medicine and the Weil Institute for Neurosciences and the Memory and Aging Center. And so Dr. Casaletto is both a practicing neuropsychologist and a researcher, and her work examines cognitive aging and dementia, specifically proteomic markers, digital biomarkers, digital neuropsych tablet assessment, actigraphy. She has a whole interesting line of research that maybe we'll hear a tiny bit about, maybe not, on endocrinology and perimenopause, menopause, working with that big study. And basically identifying biobehavioral targets for dementia prevention. And that looks like what we'll hear about today, from behavior to molecule for cognitive resilience. So please help me welcome Dr. Casaletto.
Thank you so much for that kind introduction, Dr. Kennedy, and to Ashley and all the organizers for inviting me here today. I haven't spent time at UT Dallas, so it's been really fun. So thanks to everyone that I've met, and hopefully I'll make more friends as we go through. So as Dr. Kennedy mentioned, I'll be talking about cognitive resilience. We're going to start at behavior and hopefully boil it down into the individual molecule. And the way I like to think about this is first start by the problem that we're trying to solve, that we're all trying to solve here, which is cognition with age. And so what happens to cognition with age? Well, these are real data from the NIH Toolbox Cognitive Battery of changes of cognition with age. And this is what we see. We see rapid gains in cognitive skills in early childhood, peaking around our 20s and 30s, and then, mild but definitely measurable decline as we get older. These are all typical folks. But I really think that the singular line belies the truth of the situation, which I think many also thought about, which is a ton of heterogeneity around this line. And the dots that get me really excited are these ones on the far right here. So people in their oldest age band, 70s and 80s, who are performing like people in midlife, people 10, 20, 30 years, they're younger. So what is it about them and what makes them special? How do we all keep ourselves on that trajectory? And if we take these data and actually turn to some of our community autopsy studies, we've been learning that actually, if we're lucky to live long enough into those oldest age bands, all of us will develop pathological lesions in our brain, essentially. But very, very few of us will develop dementia. And so what that tells me is that there are naturally occurring phenomenon that help us repair and rework our brain as we get older, just like any other organ system that undergoes wear and tear. And this paradox is just what's kept my interest going, that pathology may be necessary, but it is far from sufficient. And I'm hoping today I can tell you some of the other sufficient factors. And just to really take it home, I always like to show a little bit of a case example. So these are two participants in our aging cohort. We call our cohort branch. It's the Brain Aging Network for Cognitive Health at UCSF. I co-lead this cohort with Joel Kramer. And these are just two participants in our cohort. So on the left-hand side, we have an 81-year-old woman, right-hand side, 74-year-old gentleman. And if my cursor works, which it won't, but that's cool, we'll just take a look at the T1. So this 81-year-old woman, you can see that her ventricles are a little bit enlarged. Her gyri are also a little bit enlarged, suggesting that there's been some diffuse atrophy. Her medial temporal lobes have some really generous spaces. And if we look at her flare, her white matter hyperintensities aren't that much better, kind of around the ventricles, becoming confluent, punctate kind of throughout. There's some stuff happening in this brain. However, the gentleman on the right, you can see his ventricles are smaller, gyri tighter. His hippocampi are nice and plump, barely anything on T2 on flare signaling here. But what do they look like clinically? Well, this woman on the left does quite well on our global cognitive screener. She's diagnosed as clinically normal. She can recall 15 out of 16 words after a 20-minute delay on a memory test, and she's quite speedy on a trail making test. Whereas this gentleman on the right is diagnosed with mild cognitive impairment. He is impaired on our screener. He cannot recall any of the words after a 20-minute delay on our memory test, and he's quite slow. And so again, I think we see these cases all the time in clinic and in research. What is it that keeps this woman going, and how do we all get? So those are the questions I've been trying to tackle. And so when thinking about this in grad school, I was always taught when in doubt, draw it out. So you kind of draw out all your constructs and try and put them together. So this is my working model of cognitive resilience, and I just want to acknowledge that all models are wrong and some are useful. So hopefully it'll continue to evolve as we get more data. So the way I think about cognitive resilience is bucketing into two different domains, actually. One is resistance to the development of pathology, and the second is cognitive resilience in the face of risk factors and pathology. I think it's helpful to define these two groups because the resistance group is really primary prevention. How do we stop these lesions from happening? Whereas the resilience group is secondary prevention. Okay, we've developed some lesions, some changes in the brain, and how do we stop it from progressing into symptoms? And so just to walk through what this could look like, use whatever terms you want. I'll just say in the resistance, reserve, resilience, or people use a ton of different terms. All I ask is that you define them. So I'll define them here. So a risk exposure could be something like age or carrying an ApoE4 allele. And you could have an ApoE4 allele, but be resistant to the development of amyloid plaques. What drives that, right? Or you could have a brain full of amyloid, but you're cognitively resilient to manifesting the memory symptoms of that amyloid. What's driving that? And so, again, you can move in and out lots of different biologic constructs. And my goal is to fill the green and pink dots here with what are the biological factors? Can we work from a system, cellular, even down to a molecular level of what are the mediators of these resistance and resilience phenomenon? Great. It turned out it's really hard to do this, to find these resistance and resilience molecules. And so what we've done is we take a step back and think about, well, what do we know are already associated with resistance and resilience phenomenon? And so there's been decades of work thinking about behaviors that we know or factors that we know are neuroprotective over and over and over again. And so what we do is we go really deep on the biology of these behaviors and known neuroprotective factors with the hope that it'll lead us to biologies and maybe even molecules that are actually subserving these resistance and resilience phenomena. And I should mention, please jump in if you have questions. I'd love to talk this through together. So for today's talk, I'm going to just dabble in a couple of factors. We've gone really deep on exercise for many reasons, but it's one of the strongest predictors of resilience phenomenon. I'll also talk a little bit about sleep and heart rate variability, and at the end, I'll touch on some of our sex differences and health work. Hopefully I'll get through it all, but if I don't, come find me later and we can chat more. Okay, so 3 chapters. The first chapter will be the longest, I'll warn you. So we're going to talk about physical activity and exercise. So why hone in on exercise? I never would have thought I would be studying this 10 years ago. But truly, it has some of the largest and most consistent effect sizes with dementia prevention. 30 to up to 80% reduced risk of dementia, depending when in the lifespan you look. And we've seen this in our local branch, our aging cohort locally. Older adults who engage in more physical activity have better outcomes. We've even shown that this is the case in people who carry an autosomal dominant inherited genetic form of dementia. So this is in a study looking at frontotemporal dementia in people who carry genes for this disease. And we found that gene carriers that engaged in greater physical activity had slower functional decline over time and slower progression of their neurofilament light chain, which is a biomarker of axoneuronal degeneration. So again, though, my inner 3-year-old is like, that's great, but like, how is this happening? What's the biology that's driving this? And if it's so strong. So I'll take a minute. This is some older work, but after this, it's going to be pretty much all new work. So hopefully you'll kind of wade through these new data with me. But first, this is a series of papers that we did looking at some of the cellular links between physical activity and brain health using the Ross-Matt cohort. So these are older adults. They wore actigraphy devices before they died. They did all these cognitive measures. And then they went to autopsy, and we could look at their brain and think, can we start to link lifestyle to brain changes? And without walking you through the entirety of the studies, essentially what we found is that older adults who engaged in greater physical activity on brain tissue, we could see that they had lower microglial activation, so less brain inflammation in their brain. And this was particularly true in older adults who had a little bit of evidence of Alzheimer's pathology, so maybe less Alzheimer's related neuroinflammation. But of course, we wanted to look at other maybe clinically relevant outcomes. And in the same cohort, we found that the older adults who engaged in more physical activity also had better markers of synaptic health in their brain tissue, as well as better cognition over time. And so then again, we wanted to throw it all together into kind of a how of the who, so moderated mediation. And essentially what we found is that the relationship between physical activity and synaptic or cognitive outcomes, about a third of that relationship was explained through microglial activation. But the size of that mediation depended on how much Alzheimer's pathology someone had. So you can see about a third of the relationship between activity and synaptic or cognitive outcomes was mediated through microglial activation, but it was a lot stronger if they had a little bit more amyloid in their brain. So said in English, physical activity relates to better synaptic and cognitive outcomes via reduced microglial activation, especially in adults with Alzheimer's pathology. So this is sort of giving us a scoping view of some of the cellular readouts potentially of physical activity, but we wanted to get a little bit deeper to think about biomarkers and biology and treatments. So can we get down to the molecule? So in this next study, this was led by Rowan Sloaner, who's a very talented early career scientist on the team. And we wanted to ask, can we pull out molecular signatures of physical activity from blood and link them to brain aging? So again, this is in our branch cohort. Everyone wears a Fitbit for 30 days after being in the study. They also got blood draws, and then we assayed their blood from the SomaScan assay, which quantified 7,000 protein levels. And of course, 7,000 is a lot of proteins, so we do some data reduction with a technique using WGCNA, which is a weighted gene correlational network analysis. It's a fancy way of doing a factor analysis to see what proteins hang together and group together. And then you can kind of see from the groups of proteins that they have biological readouts. And so we did that in our small cohort, but then we also replicated across 2 independent exercise cohorts. So the Heritage was an exercise trial. It was a 20-week pre-post, so they got blood pre-post the exercise intervention. And then also the ERIC study, which is a midlife study of aging that you may be familiar with, who did self-reported physical activity. And the nice thing was both of those cohorts also got SomaScan. So with these proteomic platforms, we're now getting them across multiple cohorts, and we can start to triangulate data across cohorts. So 3 exercise cohorts. What did we find? And I'm just going to kind of jump into things, and you shouldn't read all this. It's just there to highlight. Essentially, from the 7,000 proteins, we could extract 31 modules or groups of proteins. And then we could do some gene ontology enrichment to figure out what's the biology. And you can kind of see the biologies that seem to be represented here. And I'm highlighting 2 of the modules here, the M9 lipid homeostasis and M12, the extracellular matrix neurodevelopmental module. I'm highlighting those because they tracked with physical activity in branch. They changed in the Heritage study and in the ERIC study. So across all 3 cohorts, we saw these groups of proteins particularly shifting. And so if we just plot them to take a look, you can see older adults with greater steps had lower levels of this lipid biology module, whereas greater steps associated with increases in this extracellular matrix module. And you can also do cell type enrichment on the proteomics within each module. And the cells that were particularly enriched were neural, oligo, as well as endothelial. But of course, this is quantified in blood. And so we wanted to ask, like, is this at all relevant to the brain? It's all peripheral right now, even if the cell types same. So then we took these modules and we started to link them into ADRD cohorts. So 3 additional ADRD and AG cohorts. And the punchline is that module 12 won, and module 12, this ECM neurodevelopmental module, it tracked with executive functioning in our local cohort, as well as processing speed and mood. Levels also tracked with, so this module increased with exercise and it decreased in AD, PD, C9 open reading frame, and MAPT, which are FTD genes, and progressive supranuclear palsy. So in 2 additional cohorts, we were able to quantify it across these diseases. So increase with exercise, decrease in disease. And then we were able to do a magma analysis and look, okay, what do the proteins in this module represent in terms of what we know about Alzheimer's gene enrichment? And it was pretty remarkable, but hopefully you can see that this module 12 really won in terms of representation of genes that we know are involved in Alzheimer's disease. So it seems to represent something particularly relevant to the brain. Okay, can we dig more into this module? So this module comprised 165 proteins, and you can see, well, you can't really see the lines, but there are lines kind of connecting the strength of the relationship among the proteins. These are the hub proteins, so the most connected proteins here. And then we can start to ask questions, what are the proteins in this module that are most important? And so we just started mapping them across the diseases that we're looking at. There are several that were really interesting, but I'm just going to highlight one here called ANTXR2, or Androxin toxin receptor 2. And what we found over and over again, this is a protein that increased with exercise. It decreased in AD, PD, C9, progranulin, MAPT, and PSP just consistently over and over. And then we looked at it in ROS-MAP, and we found blood levels of ANTXR2 predicted cognitive decline over time. People with higher levels, slower cognitive decline over time. And then we were able to do something cool in ROS-MAP, again, because they have brain tissue. What do blood levels of ANTXR2 map to in terms of brain proteomics? And we found that ANTXR2 elevations tracked with things like synaptic potentiation. I mean, these are exactly the factors that we think about and want to see. So hopefully, I told you the story of how we can narrow the exercise down into an individual molecule. But more broadly, I think this is an exciting way to leverage known risk and resilience factors to identify new targets for dementia prevention in this space. Hopefully I've convinced you that there is a signature of physical activity. We can replicate this over cohorts, and we see that that's quite robust. I think the ECM is super interesting. We'll put a pin in that. We're going to come back to it. Maybe this is a resilience target. And maybe there's an individual molecule that, again, is starting to boil down the neuroprotective components of exercise, and there are others in there. But maybe this could be a biomarker or readout of understanding, is my exercise good enough for my brain health? Can I track my ANTXR2 to make sure my exercise is working for my brain health? So we're always looking for folks who want to play around with molecules and animals. So reach out if you know anyone. And I'm just going to take a minute on the ECM because I'm going to come back to it. Who here has heard about the ECM? Oh, a little bit. Nice. Okay. I had not thought about this at all. So I did a little bit of reading. But this extracellular matrix, essentially, yeah, it's this macromolecular network of proteins that kind of hangs out and provides structure to all of our organ systems, including the brain. And in the brain, it actually comprises 20%, a fifth of the volume of the brain. And essentially, it provides this matrix so that the synapses and other cells can connect where they need to connect, retract when they need to retract, and kind of have that structural plasticity that it needs, as it more nicely says here on my bullet points. But I think it's really kind of the structural correlate of plasticity in the brain, which is really interesting to think about. And there's precedent. So now that there have been multiple proteomic studies in brain tissue, the ECM and the matrixome has come up as one of the earliest biological shifts in Alzheimer's disease. And not only that, but if folks are familiar with the ApoE Christchurch mutation, this is a genetic mutation that confers cognitive resilience. actually confers resistance to the development of tau and cognitive resilience. And it was found in people who carry presenilin-1 mutation. So if they also have this mutation, they don't develop the cognitive symptoms despite having an autosomal dominant form of AD. And the really cool thing, they're still working out how this amazing gene works, but it's a heparan sulfate polyglycan, so it's an ECM protein. So I think there's converging lines of evidence that the ECM may have been overlooked and could be a resilience target. Okay, so I took you kind of from behavior down to molecule, and now we're going to go the other way and talk about TIM2, which, spoiler, is also an ECM protein. So this is a collaboration with Joe Castellano, who's a neuroscientist at Mount Sinai. And he had this beautiful Nature paper, maybe some of you have seen it, building on the blocks of heterochronic parabiosis, this idea that you can take young blood, put it into old mice, and rejuvenate their brains. It's amazing. And so he wanted to take the youngest possible blood. So he took human umbilical cord plasma, and he injected it into old mice. And essentially what he found, all you have to see is that there's more white dots here, there was more neurogenesis and behavioral benefits to the umbilical cord plasma in the old mice. And of course, what is it about the plasma? So he did all these analyses and essentially came up with TIM2 as one of the top factors that was upregulated in this umbilical cord plasma. He also showed that TIM2 crosses the blood-brain barrier, so from blood to brain directly. And that if you just take TIM2 alone, you can recapitulate some of the benefits of this young blood. So better LTP and memory in these mice. But I study humans, so we wanted to ask the question, is this at all relevant to the human condition? And is TIM2 modifiable? Is this like a modifiable rejuvenation protein? And so this was a story that Emily Paolillo, another very talented early career scientist on the team, took on and asked this question. And so essentially what we found, just really briefly, is in our local cohort, higher levels of TIM2 associated with better cognition in our healthy agers. And of course, we never trust one cohort, so we turned to our friends at Stanford and Rush, and we found, in fact, in their cohorts, higher levels of blood TIM2 correlated with better cognition and slower cognitive decline longitudinally. So there is relevance in the human. I should mention she also found this on brain structure, so larger gray matter volumes. And then she took our Fitbit data from our older adults and found, yeah, not only is TIM2 related to cognition, but higher step counts tracked with higher levels of TIM2 in our older adults. And again, we turned to our friends at Ross Map and found the same thing. So we turned to Joe and we said, this might be a modifiable protein. He's like, great, let's test it. Like, we can't do this in our observational human studies. And so he built out an enrichment paradigm for his mice. And essentially what he found is that indeed, if you put mice into an enriched environment, you can increase their peripheral levels of TIM2. And if you knock out TIM2 and put mice into an enriched environment, they don't get the rejuvenation effects as much, which means that TIM2 seems to be mediating these enriched environmental effects on the brain. So yeah, what is TIM2? What am I talking about here? So TIM2, it inhibits these MMPs, which you may or may not have heard of, but again, involved in the ECM breakdown and buildup. We know it crosses the blood-brain barrier, which is really exciting as a therapeutic, and it's produced peripherally, but it's also produced locally in the brain by neurons, which is interesting to think about. We know TIM2 has a binding partner with MMP2, and there's some data that suggests that essentially these two work together hand in hand to limit too much ECM, which can be too rigid and not flexible enough, which promotes plasticity if it's nice and smooth. Joe has also shown that TIM2 decreases microglial activation. There is also some hints that maybe it helps support blood-brain barrier functioning and bad angiogenesis that can happen as we get older too. So more to come in that space, but again, hopefully an example of how we can go back and forth across the bench, across behavior to molecule. Okay, so really cool molecular studies. We give these talks in the community. And like, this is the question we always got, like, that's really awesome, but like, really, what should I be doing? So this is work led by a postdoc on the team, Claire Cadwallader. And she set out to ask this question, which is, really, what type of physical activity is best for the brain? I think that that's not quite clear yet. And the pragmatics of it are like, you cannot possibly ever run a randomized controlled trial of every type of physical activity for brain health. There's just so many issues with RCT for exercise for dementia. So can we get a little bit more creative about this and leverage our Fitbit monitoring? So again, folks wear their Fitbits for 30 days. It's blinded, so it has no pinging or activity happening on the Fitbit. They just are asked to do their regular movements and go out in the world. So Claire really wanted to develop an algorithm in order to isolate when are there activity sessions, or can we define exercise sessions in the real world? And then from those exercise sessions, can we pull out the features that are best for brain health? And can we even start to look at person-specific factors like age and sex, which are much harder to do in the clinical trials? So I'm just going to walk you through how she built out her algorithm. So this is an example of a 12-hour day. This is someone's actigraphy data itself. And so first, we wanted to define when someone started a session. And so we wanted to keep the bar low. These are older adults. So we did this by saying anytime someone's engaged in 40 steps or more for 10 minutes or more, a session was started. So that's sort of the threshold to hit. But, you know, people take breaks, they go for a walk. Oh, you can't really see this, but, you know, they might tie their shoes. So we wanted to allow breaks in between there. So people can have breaks up to 9 minutes and it still counts as their session. And so essentially what's in green here is a session. And what's not in green is out of session. And so from there, we can derive things like what were the types of movements, a little blurry, but intensity, duration, lots of different features of the sessions themselves, as well as out of session steps. And this is how we're and this was just sort of a gut check, but we defined an exerciser as anyone that engaged in one or more sessions in the 30 days. You know, that's a really low bar for an exerciser, but it was the way we decided to move forward with it. So this was a good gut check that you can see that most steps are taken out of session, as you would expect, right? But most people's vigorous steps were taken in session. And so that means, okay, we think the algorithm is working. It's defined it this way, but good to see. And then from here, she built some really fancy models that I'm not even going to pretend to go through, but these are ridge regression models that are machine learning models. And essentially, she entered in 42 different features from these sessions and out of sessions. We included interactions between the different features. We included interactions with age and sex to ask questions about person-specific features. And then we looked at our kind of standard outcomes, cognitive brain structural outcomes. And what did she find? Well, of the 42 features, we're kind of zoomed in on the top here because 6 out of the 42 were the most important across all the outcomes. Like really none of the other ones played out, and it was really quite consistent. So the most important feature of physical activity for these brain health outcomes was frequency. So engaging in more frequent exercise sessions was the most important, strongest relationship across these outcomes. The second was cadence or intensity of movement. And then, you know, duration was like a little less important, which I was kind of surprised by. And then interestingly, we saw a bunch of sex interactions. So these show us that females seem to show a stronger relationship between those parameters and brain health outcomes. And I'll just say, I'm not showing this, forgot to mention it before, but if you even engage in one exercise session, those folks have better white matter health in our group. But then above and beyond, within the sessions themselves, you can start to see some of these individual features. Okay, and so kind of lessons learned from this one, for me at least, is that sessions, even of low intensity for as little as 10 minutes, are still associated with better white matter outcomes, as I just mentioned. And we think that maybe these shorter sessions that are of higher frequency and maybe a little bit higher intensity might be the best for the brain, getting like these pulses throughout the week and the month. And I think this is really interesting because it parallels nicely with survival data. So there have been a couple of papers out on mortality and survival and exercise, suggesting that like these high intensity interval training they are particularly associated with survival. Probably anything good for survival is good for the brain. So maybe we're seeing some parallels with this. Also really interesting that females may have more to gain, particularly with session frequency and duration. We have some data that suggests males show some benefit, some data that suggests females show more benefit. The field is really mixed here. What that tells me is there's probably a third variable that we're missing. And so I think trying to understand this will be really important and quite pragmatic in terms of giving recommendations to the population. And I'll just mention, even in the non-exercisers, we looked at step count and brain health outcomes. And even in them, we found that step count tracked with better brain health outcomes. So the moral is like any movement counts, but like if you're gonna get precise, yeah, maybe do it like a little more frequently and if you can a little bit more intensely, rather than doing these long epics. And I should mention, we didn't see any interactions between the session features, which means you can't be a weekend warrior. You can't make up for your frequency with more intensity or more duration, right? Like the body wants, the brain wants this more regular cadence. So to summarize some of the physical activity work here, I think what we're starting to see is physical activity, maybe particularly high frequency, can track with some of these signaling in the periphery, these blood factors. I showed you a couple of them. We have other ones that we're interested in. I think that these factors in the blood may be particularly related to structural remodeling, maybe especially in the periphery to blood vessel remodeling. So maybe the ECM of the blood vessels I've been thinking a lot about, but also maybe in the brain. And from there, we can see decreased inflammation in the brain and better synaptic outputs. So this is sort of my working model of what I think might be happening with physical activity. Okay. Physical activity. That was chapter 2, chapter 1. Any questions on physical activity? Yeah.
That's great. I'm just curious about the effects of duration. I wonder whether it's related to the fact. So, how was communication confidence like entire day, or was it the duration of like each like 10-minute or more second? Like how long is that second? So, I'm just curious about like that. How it's calculated could be the reason why the frequency, like duration, is not showing that big of an effect.
Yeah, and I still think there are features we're missing. So I really love this question. So duration was calculated as how long was your exercise epic session, and so 10 minutes was a minimum, but it could be as long as they exercise. So yeah, it wasn't out of session. It was just in session.
It's not across the day or.
Yeah, we've looked at time of day. We haven't been able to pull out much, but I think we need to look at it the right way. Yeah, I think there's lots of other features that this was a first pass that we can still learn from. Okay, so this second chapter, I'm going to talk a little bit about rhythms for brain health and some, again, new data coming out. So thanks for your patience as I go through this. Sleep, we know this is another neuroprotective behavior, known neuroprotective behavior. Many studies that show better sleep duration, lower dementia risk. We can see this for decades out. And yet sleep is not a part of the Livingston Lancet 14 modifiable factors for brain health, right? Why? It's kind of complicated, especially sleep duration. You can almost see this hue that's happening in the error bar there. So maybe with duration, we're not quite getting at the right feature. And in fact, again, looking at the mortality data, let's look at mortality for brain health. We see that there was a recent paper that came out that showed that actually regularity of sleep, so how regularly you go to sleep at night and wake up in the morning, is a stronger predictor of mortality than duration. And so we wanted to ask the question, okay, maybe is that true for brain health as well? And again, this is led by Emily, and she wanted to ask this question, is regularity really the special sauce that we've been missing? And how does it relate above and beyond duration? So this was in our aging cohort at UCSF, healthy folks. They do all of our classic measures, and then 30 days of Fitbit monitoring. Average age about 70. And I'll just walk you through this sleep regularity index. So essentially from the Fitbit, we can get their sleep regularity. It's sort of beautifully normally-ish distributed. So you can see what that looks like here. And just to hone in, these are real participants' data. So someone with a low SRI, you can see on the x-axis, that's time, 12-hour day. And you can see like the low SRI person is having a lot of awakenings throughout the night. And this is in contrast to a high SRI participant where they have really tight, nice sleep and throughout the 30-day monitoring period. And just to highlight, we think SRI is capturing something unique. So SRI really did not correlate with average duration. It did correlate with variability in duration, but it still is only capturing about half the variance. So we think SRI might be capturing something additional above and beyond these kind of traditional features. Yeah.
So it's quantifying sleep onset, not the fragmentation.
It's quantifying onset and wake. So just the time at which you.
But the gaps in between.
No, not getting
fragmentation.
Wait, so no, none of the fragmentation. So yeah, that's a great question. Okay, so we took a peek at this SRI index, and again, we covaried for average duration asleep as well as variability asleep, and then all the typical covariates. And I was kind of shocked, but we saw that SRI tracked with cognition, it tracked with mood, it tracked with gray matter, it tracked with white matter. Like it tracked with all the outcomes. I just feel like very rarely do we have a modifiable risk factor that pulls in all these orthogonal outcomes. And it was just so consistent. So I'm like kind of convinced by this, trying to change my behavior, but we have more to do here. But I just thought I'd sort of show you where some of our early data are. Though I will say, I think duration still matters. So we looked at plasma p-tau levels, and these tracked with duration, but did not track with SRI. So maybe there's different paths to like typical aging versus an Alzheimer's path, depending on your sleep features too. Just to put that out there. Please, yeah.
So it's an interesting relationship between SRI and all the other measures. Is that controlling for, I don't remember the age of the sample.
About 70.
Is it controlling for person age, and maybe more importantly, is it controlling for like total sleep time?
Yeah. So in these models, it controls for age, sex, education, total sleep time, variability of sleep time, and then TID.
Oh, sorry.
Yeah, no worries. Yeah, that's what I thought was so cool. And in those models, duration and variability in duration did not come out as significant predictors. It was just SRI once you controlled for that. So it was pretty analytically convincing to me. So SRI has been, a new metric that we've been toying with. And then another new metric we've been thinking about is this heart rate variability. So heart rate variability is the variability in your consecutive heartbeats. And it's thought to be a marker of both autonomic and cardiac function. So it's sort of an aggregate of the sympathetic and parasympathetic input from vagal nerve, which is incredibly important for brain health. We know it declines with age, and this is always a little counterintuitive, but you want high heart rate variability. So having higher heart rate variability is thought to be like a little more adaptive and flexible. You can kind of think on your toes and be resilient physiologically, whereas low HRV is thought to reflect maybe an autonomic imbalance, maybe too much of that vagus nerve or too much sympathetic activity. And what really got me excited about HRV is that it's incredibly modifiable. So anything that we can modify in a human, I'm totally game to figure it out. So we know that it's modified by breathing, by yoga, exercise, stress reduction techniques. So here we might have a nice access into the brain via HRV. And so I'm just going to show you just a tiny bit of the prelim data. Oh, before I get into that, I'll just mention whenever we have a new metric that we're looking at in a Fitbit or really any device, especially a commercially graded device that are a little bit more black box, we try to validate them. And so we paired with one of my collaborators at the Memory and Aging Center, Virginia Sturm, and she does in-lab physio. So she kind of like puts electrodes on the whole body and does all these amazing studies on physiology and emotion and aging and neurodegenerative disease. And so we asked her, would you mind slapping some of your physio on our participants as well as when they do the Fitbit and see if we can really track HRV with high fidelity. And so essentially what we found in lab, it's called respiratory sinus arrhythmia. But essentially what you can see here is that FIPID-derived HRV and in-lab-derived HRV tracked up quite closely with one another. Not perfect, but a pretty good correlation. Kind of what we see across like our cognitive measures. And it was in a mixed dementia sample. So we think it might be a pretty good measure of HRV based on these pilot data. And then really interestingly, in our healthy aging cohorts, if they're normal folks, we found that higher HRV tracked with more step counts or more exercise, lower vascular burden, greater P-Castle brain perfusion, and lower white matter hyperintensity. So like a really strong vascular signal coming out of HRV. So we think this might be kind of a cool metric to track and think about moving forward, but just at the early stages. So thinking about these sort of rhythms of our body and how they shape our brain, I think this SRI index is really convinced me it's quite relevant to brain aging. Maybe duration of sleep is relevant, but maybe for other pathways, as I mentioned, maybe more this Alzheimer's pathway, more maybe it's tracking more with like WASO, these fragmented sleep phenotypes. That might be just simply different biology. And HRV, we have some now early data suggesting it could be a good indicator of these autonomic and cardiovascular changes that are potentially quite relevant to the brain and intervenable, which is always exciting for me. But mostly, hopefully, I've convinced you that we can use actigraphy monitoring, even commercial level, like Fitbit devices, to get much more precise about how we give brain health recommendations moving forward. And I'll just acknowledge these are all observational data, so we have no idea the directionality. So it's possible that changes in sleep-wake cycle are a marker of brain health, or that someone's about to shift into disease. And if you think about circadian rhythms and shifts in circadian rhythms, it certainly could be that it's headed in another direction. But in that case, we can really think about wearables and sensors in the home as these scalable tools to potentially catch people at that very early phase and monitor and flag when they're at risk. So I think we can kind of go where the data support. Okay. All right, hoping to wrap up really soon, but I'll just touch on a little bit of our work in women's health. This has been a really fun space. Female biology has taught me a ton about cognitive resilience. So we know that dementia manifests differently by sex. Specifically, we're seeing that females carry a higher neurodegenerative burden. So what we see, and this has been shown in many studies, is that for their level of amyloid, women carry a higher burden of tau. And this has been shown with PET, the CSF, with autopsy, over and over, longitudinal, over and over again. We also recently showed that this was the case in Lewy body disease. So if you control for amyloid, women with Lewy body pathology in their brain also have more tau compared to men. And then recently we showed this was the case also in genetic forms of frontotemporal dementia. So women who carry a gene for FTD, this is a totally molecularly different disease, right? Not caused by plaques and tangles. We found that women gene carriers had steeper rates of brain atrophy on MRI and NFL trajectories over time. So there's this degenerative phenotype in women. And yet, we're seeing that women are outperforming and outliving this neurodegenerative burden, which is incredibly fascinating. So I just showed you this plot of these participants in our ADRC, and this is what their brains look like. But if we look at their clinical functioning, hopefully you can appreciate that the blue and teal bars are not different from one another. So despite the fact that the women are carrying that higher tau burden, we are not seeing it on our favorite functional and memory measures. And then this was a recent meta-analysis that came out looking at predictors of survival in AD. And they found that sex was one of the strongest predictors of survival. And specifically, actually that male sex was one of the strongest predictors of time to death. And hopefully you can appreciate that the effect size of male sex was as strong as things as having cancer or a movement disorder. So these are not small effects. We're really seeing them. And then, as I mentioned, we did a study in genetic FTD, and we found that in the gene carriers who were not yet symptomatic, that women essentially outperformed their NFL levels. So in the red there, those are people with high NFL, And you can see the women are progressing more slowly on cognitive outcomes. I mean, of course, this begs the question, how and why is this happening? Starting on more the risk side, we've been thinking a lot about menopause. So we know that the symptoms of dementia manifest in our 70s, but actually, as you all know, got to turn the dial back. The pathology is manifesting 10, 20, maybe even 30 years earlier. This puts us perfectly in the menopause transition at 51. And actually, if we look at community autopsy studies, these are healthy folks looking at AD pathology across the age range from the Brock series. It's a really cool paper. But they did a sex analysis, and what they found in purple here is that females started manifesting Alzheimer's disease changes in the brain earlier than men. And actually, if you look at the age range, it's right around the time of menopause. So a coincident midlife window. And then work from my friend and colleague, Rachel Buckley, has shown that compared to premenopausal women, postmenopausal women show this sort of elevation in tau, especially for their amyloid level. So maybe this is a window of trying to understand kind of the fundamentals of disease pathophysiology. And we've done some work looking at timing of menopause with tau. And essentially what we've seen, and actually this is from Rachel's group too, is that earlier age of menopause tracks with more tau, more dementia risk. So this is in orange, you can see that the women who underwent premature menopause had higher levels of tau for their amyloid level. We found that this was the case in the Lewy body study as well. So it was really that tau effect was really driven by women with earlier ages of menopause. And actually we see it in our local branch cohort. These are healthy women, and we can see that p-tau, plasma p-tau tracks with their age of menopause. So there's something about age of menopause that might be giving us some hints into this biology. And so just taking a step back, this is a perspective that Dina Duvall and I have been working on, and just kind of trying to put it all together, that women have this paradoxical increased risk for decreased resistance to the development of neurodegenerative disease, but increased cognitive clinical resilience in the face of neurodegenerative disease. And so this is how we've been trying to kind of put it together and think about, can we fill this gap? What is it that's driving this curve? And can we really fill this gap, right? This is therapeutics. What is it that women have that help them survive and have better memories and do so well with these pathologies? And we've been thinking a lot about the X chromosome in that context. Okay, so just to summarize, hopefully I've convinced you that risk factor research really gives us the fundamentals of dementia. Dementia is complex, context is so necessary, and pathology is just not enough. It is not sufficient to develop dementia. All of these other features are so incredibly important. I also love risk factor research because it gives me immediate gratification, things that I can do now, things that I can bring to my patients to do now, but it also sets me up for the future, discovery work. What are the biologies driving this? Can they lead to new biomarkers and therapeutics? I'm very excited about this space of women's health and female biology. I think this is just a treasure trove, historically understudied. There's so much to learn here, and it is low-hanging fruit. So if you're interested, now is the time. And really more broadly, my vision would be to develop biologically and molecularly driven behavioral interventions for dementia prevention. If we can prevent half of dementia with simply modifiable risk factors, let's do this on a really precise level. Let's get at that biology for that person and make our interventions as potent as possible. So with that, yeah, just a huge thank you to participants and families that give me my job. The team is awesome. They're so much fun. So huge gratitude to all of our collaborators and funding sources. And then if you're interested, I direct the biomarker core on the Bowers Women's Brain Health Initiative we kicked off just about 2.5 years ago. Check out our website. We have lecture series, and then we're launching a menopause study next month. So it's a 3-site study to really understand what happens to the brain during perimenopause. Thank you.